Hello everyone, welcome to OK Biology Classroom and welcome to our video lesson 4.3. Here we are going to look at DNA replication, but lesson 4.3 is going to be in two parts. 4.3.1 is going to look at the DNA replication process in a simplified form. Then there will be 4.3.2 that will be looking at the same process but in an expanded form. Okay, so in 4.3.1 here we are not going to talk about the specific enzymes involved, and as well we are not going to talk about the work of the primers, but we'll be adding all of that in our next lesson video. That will be the 4.3.2. And as a prerequisite, I would like every student who wants to be part of this particular lesson to have good knowledge about the structure of DNA. So if you don't have good knowledge about DNA structure, please watch our video lesson 4.1 which deals with that before you proceed to watch this particular video, because this is going to depend a lot on the structure of the DNA. But before we get into what we have for today, please remember to subscribe to our YouTube channel so that you always have our videos whenever they are released. Thank you. So DNA replication process, and we are saying that at the end of this lesson, the student would be able to describe the major stages involved in DNA replication. So like I said, in this case, we are going to have a summary of it, or a simplified form of it. So we are picking the main stages involved in DNA replication and talk about them. Good. But remember to watch the next lesson video so that you will have the additional information there too. So a simple definition for DNA replication: it is the process of making a copy of DNA molecule. So just like in our case, we are able to artificially make photocopies of information that are important to us. Within the cells, photocopies in quotes are also made of DNA molecules. And so the point is, why would a cell have to make copies of the DNA molecule? DNA replication always occurs before cell division. And the reason is that new cells are going to be formed, and each of these new cells would need to have the same DNA information as existed in the original cell. Good. So that's why we say that this provides each new cell with a full DNA molecule, and for that matter, full chromosomes, because you know that the DNA molecules, they make up the chromosomes. Good. As additional information, DNA replication occurs in the nucleus of eukaryotes, but in prokaryotes it occurs in the cytoplasm. And just like I said, after DNA replication, two molecules of DNA would be formed. To make it simple for a student, I'm going to talk about the DNA replication process in three stages. The first stage is the unwinding and unzipping of the DNA molecule. The second stage would be the formation of new strands. Then the third stage would be the zipping and winding of the DNA molecules. Okay, so now let's start with the first stage. Well, from our video lesson 4.1, where it talks about the structure of DNA, we got to realize that the DNA as a molecule exists as helical structure, having two strands that have been twisted about each other. So like we are seeing in this case, this is one strand and there's another strand, and they have been twisted about each other. But before replication occurs, there is a need for this helical shape to now be straightened up. And in doing that, it would have to unwind first. So you see unwinding occurring here. So note that in the first stage, the DNA molecule would have to unwind. After unwinding, the next step is that it needs to unzip. And unzipping means that the hydrogen bonds that were holding the nitrogenous bases would now have to be broken. And as they are being broken, then it leads to the unzipping. So it's just like opening up the zip of maybe a bag. Okay, so the hydrogen bonds are broken in this case, and then it is unzipping. Remember that the unzipping is going to occur along the entire length of the DNA molecule. So that is going to lead to the formation of two separate strands. So this is one strand and that's another strand. And these strands have been formed because the unzipping occurred along the entire length. Okay, so it's so simple. Now on the next slide, I'm going to show these same separate strands. Right, this strand and that strand, I'm going to show them on the next slide. But this time I'm going to give details about the nitrogenous bases. So don't be confused. It's the same thing you are seeing here, which I'm going to show on the next slide. Just that I'm going to show specifically what nitrogenous base exists here, right, and as well on this strand. So let's look at them. Yes, so now they have been unzipped, as you see clearly here. So this one strand and this another strand. So these are the nitrogenous bases that are present along each of the strands, right? And you can see the base pairing rule working here as well. Okay. Now each of these strands is going to serve as a template. So that means each of them is going to serve as the original against which the new strand would be made. So this is going to be one template and this is also going to be another template. So one strand is going to be formed against this template and another strand would be formed against this template. So that's the next stage. So let's get into that. Formation of the strands, stage 2, and look at how that occurs after unzipping. Formation of the strands. Okay. Within the nucleus of the cell, so in this case we are focusing on the eukaryotic cell where replication occurs within the nucleus. Note that within the nucleus of the cell there are many free nucleotides. In this case I'm showing them here with these few. Remember that I have shown them with a few nucleotides because of space. So like what we are seeing here, these are free nucleotides within the nucleus, and I'm also showing some here as free nucleotides within the nucleus, but there are numerous free nucleotides. So these free nucleotides are now going to be arranged against the strands according to the base pairing rule. So in this case where you have A here, a T nucleotide would now be positioned against it, so in a complementary way. So let's look at it. So a T against A, G against C, T against A against T. And we have talked about this base pairing rule in our video lesson 4.1. So I know a student learning with us at this stage has good understanding about that particular concept. Okay, so to continue, nucleotides are going to be arranged against the strand. So in that order, they are going to be arranged. Good. Just like we have in the case of the first strand, we can see the upper strand in this particular case, how we would refer to it. And I'm just referring to it as an upper strand because we find it at the upper region here. Now, a full strand has been formed here against the upper strand. So there are going to be two strands in this case. Similar thing is going to happen to the lower strand. So let's look at that. So they are also going to be paired along the entire strand. Yes. So you realize that in this case, you're going to have one old or original strand now having a complementary strand, which is a new one. So it happens in this upper strand case, and it happens as well in this lower strand case. Good. So that is for the stage of formation of the strands. Now, after the strands have been formed against them, the next action is that they need to zip up. So they need to be connected again. Right. So let's look at how that is also going to occur. So the two strands, one new and one old, are now going to be joined at their nitrogenous bases by the hydrogen bonds. Remember that hydrogen bonds were broken during the unzipping, but now hydrogen bonds need to be formed again. So that's what I refer to as a zipping. Good. So this is the upper strand and the original strand in that case, which had a new strand formed against it in this way. So now you see clearly that the new strand has now been bonded to the original strand. Same thing will happen to the lower strand, where the new strand would also be bonded to the original strand. Right. And that is going to lead to the formation of two molecules. So after the zipping, in that particular case where the hydrogen bonds would now connect them again, then there should be the final stage of winding them again, because I talked about zipping and winding. Yes, so they wind again into the helical structure, because that's supposed to be the normal shape or the normal structure of the DNA molecule. Remember that now we have two of them. So in the case of the upper strand, we had one old strand and then one new strand, and these two are now going to be twisted about each other again. The same thing happens to the molecule that was formed at the lower region, and they are also going to be twisted to form a full DNA molecule. So let's look at that. So the one at the upper region forms a DNA molecule, and there is another DNA molecule formed in this case as well after the winding. Good. So that is how the process of DNA replication occurs. So simple. In 3 stages, it is good to go. The first stage being unwinding and unzipping. Then after that, new strands are going to be formed using the free nucleotides. Then there will be zipping and winding to end the process. Yes. So in summary, what I said is that during the DNA replication process, there is unwinding and unzipping of the DNA molecule. Then free nucleotides would assemble along each strand. Then the 2 strands are joined at their nitrogenous bases. That is where they zip up again. And then they wind to form 2 new helical DNA molecules. So it's very simple. Alright. So just like I said, this is video lesson 4.3.1. In the next lesson 4.3.2, we are going to look at the same DNA replication process, but this time in detail. So we are going to look at the role of primers, the role of enzymes, and as well the ordering of the nucleotides during the arrangement. Good. So make sure you watch this second video as well to augment your understanding of the process of DNA replication. Yes, these are some few questions for us, and I will attach the email address as well as the WhatsApp number so that you can send your answers to, and yes, we will respond to them. Okay, so the first question is, describe briefly the DNA replication process without including the work of primers and enzymes. Then, how many DNA molecules are formed at the end of the replication process? What does unzipping in DNA replication mean? So these are very easy questions which I know every one of you can answer. Okay, thank you for being part of today's lesson. Please remember to subscribe to our YouTube channel so that you always be notified whenever our lesson videos are released. Okay.